as the last speaker, thanking the organizers for this wonderful 2 days, an incredible amount of work. On the one hand, you didn't have to get us all to Lisbon and put us up, but on the other hand, you had the enormous challenge of how to put together a conference during the period of COVID, finding ways that would all work for all of us. And thank you for everyone else for putting up with the fact that I'm so far away from the rest of you that you needed to have a late afternoon, evening conference. Coming last, I can draw upon the fact that many of the themes I was going to develop have already been introduced by other speakers. There may be some different twists on some of those themes in the way I move. The one thing that will probably be distinctive of my talk is that most talk about mechanisms has emphasized what goes on inside mechanisms. My focus today will be primarily on what goes on outside of mechanisms that affects the behavior of mechanisms, and in particular, the way they relate to organisms. So the outline for the talk, I will give a very brief comment on the standard new Mechanist account. I start there because I'm going to in various ways depart from it or expand upon it, but I also can draw out a couple themes from that account that still permeate the picture that I'm going to present. Then I'm going to start to focus on organisms, emphasizing first that organisms require what I'll refer to as production mechanisms that perform work that maintain the organism. But then I'm going to flip that around and focus on how production mechanisms depend upon the organisms in which they exist. I should have commented, I'm focusing primarily on biological mechanisms. So I'm not going to try to worry about how this plays out in other domains immediately. I will then turn to control mechanisms that are important for maintaining the activities of production mechanisms so as to sustain organisms, and finish with a focus on how control mechanisms are organized and what's the significance of the fact that they aren't organized hierarchically. Starting with the traditional new Mechanist framework, helps me to always try to ground myself in real examples from biological science. So I will be throughout this talk grounding myself in understanding of how muscles work. First, Mechanist accounts typically begin with the identification of a phenomenon. In this case, the ability of organisms to exert forces, mechanical forces on their environment, and then turn to trying to identify the responsible mechanism, what Richardson and I long ago called the locus of control, the muscle itself, but then to proceed to decompose the muscle, in this case, first into muscle fibers, but especially into individual microfibrils, and within them, the sarcomeres in which actin and myosin filaments are sliding past each other, causing the contraction. I should note, I won't say a lot about it, but I will say a lot about organization. But a further task is to show that how, when organized in the appropriate way, the mechanism is able to generate the phenomena with which one started. Okay. The move to take mechanisms apart, to find their constituents and characterize their constituents, is in many ways a reductionist move. And it seems ever more reductionist when one realizes that that step can often be iterated. The components of mechanisms are often themselves mechanisms that can be decomposed into additional mechanisms until at some point the research bottoms out. I'll have something to say about the choice of where bottoming out happens in biological explanations. But But already one then starts to see, and Stuart really developed this much better than I'm going to now, that one sees a form of emergence. The mechanism is performing activities that none of its individual components are able to perform on their own. So something is produced in the mechanism beyond what was available in the components. What's the key to that? It's the word is organization. That the parts are organized, they're coordinated with each other. That enables the mechanism to do more. It's been alluded to. The same thing applies in the case of human inventors. Human inventors don't start with don't create new basic entities. They take already existing components and they come up with something new. They can get patents for it, not by new components, but by how they put them together. It's the organization that carries us from components to what they can do as an integrated whole. And somebody used the word integration. I think that's actually very important. It's a common perspective, I think, to think that mechanisms are just sort of linear compositions of their components, but that would never give rise to a really distinctive thing called a mechanism. It's typically because of the way they're integrated by non-sequential modes of organization. I've already alluded to it. There can be multiple stages of decomposition, which gives, and since the relationship between the constituents and the mechanism is compositional, one ensures that it forms into a hierarchy. The constituents of one part are composed into that, which then becomes part of a larger mechanism. The mechanism is at a higher level than the constituents. Yet there isn't a neat organizing of these levels. The term level is often bandied about in these contexts. But one thing I want to emphasize is even when you focus just on part-whole relations, how you identify levels depends upon the way you decompose the mechanism. If you decompose this mechanism into A, B, and F, well then F is at the same level as A and B as a component of the whole. But if you don't organize them into one unit, but treat them as separate components, well, then you would give a story in which there are 5 components of the mechanism, and these components would be on one story, they're at a still lower level. At this level, on this story, they're at the same level. So also a further problem is that if we started decomposing another component here, C, there's no reason to assume that those components are at the same level as the components of of B. So we're not even giving rise to a univocal account of levels. It's highly localized on this account to the mechanism from which we started. So the Mechanist project is not per se involved in trying to reduce anything to any lowest levels of nature. The decomposition and lower levels is part of the strategy for gaining explanation. So now I'm going to turn to the relation to organisms, which have not figured very much in Mechanist accounts, the accounts of the new Mechanists. Sometimes one looks to the organism to identify the phenomenon, but that seems to be well, that's one place they might appear. Sometimes one will test the correctness of a mechanistic account in terms of whether it accounts for behaviors of the mechanism, of the organism. But by and large, the organism, I think, is often just set aside as a collection of mechanisms, and the focus remains on the mechanisms themselves. I think this is a serious neglect in the Mechanist literature that we really do need to take seriously if we're going to understand biological mechanisms, how they're integrated into organisms. And I'm going to start with the side that they contribute to the organisms. So the organisms need mechanisms. As Alvaro and others have already emphasized, organisms are themselves far from equilibrium systems. They're at low entropy. In open systems, entropy increases. What that would mean is that the organization, often very delicate organization, can break down. And if this were the case, it would normally lead to the elimination of organisms. Yet we have before us a lineage of organisms on this planet that's began 3.5–4 billion years ago from one initial living organism. This requires that each organism procures matter and energy from its environment, utilizes the matter to construct its own bodies, and then must expel the waste products. Now, it is the case that all organisms eventually fail to maintain themselves and die, but along the way, many reproduce, resulting in lineages of far from equilibrium systems. And these lineages synthesize new organisms. Fortunately for us, this process is often noisy, so that over time has resulted in some organisms becoming more complicated. We shouldn't get misled by this. There's not a directionality to evolutionary processes, but along the way, we've produced organisms that are even further from equilibrium, and then even more delicate and requiring ever more work to maintain themselves. Now, I'm going to start to modify the standard new mechanist account by introducing a couple of terms that haven't been common in it. The first is work. New mechanists typically talk about mechanisms accounting for phenomena, but not as engaged in the performance of work. And a second component, Alvaro brought it up in part of his talk, the absolute essential requirement for finding sources of free energy from which the work is generated. Jason Winnig and I then, in reflecting on these components, proposed an alternative characterization of mechanisms in terms of constrained release of free energy so as to perform work. Now, this maps in part onto the more traditional new mechanist account. The entities or parts of the mechanism are what are doing the constraining, and it's the constrained release of free energy that actually accounts for both the individual activities within a mechanism and the overall activity of the mechanism. Using Nancy's concept of capacity, due to the way it is organized, a mechanism has a capacity to perform one or more activities when free energy is released. I'm going to be emphasizing in a bit the more activities. But let me go back to my example of muscle contraction to show some of how this story plays out. I earlier introduced the idea that muscle contraction involves the sliding of filaments, actin and myosin filaments past each other. Well, it's not just an innocent sliding. The myosin filament actually is exerting a force on the, well, you can see it here, is exerting a force on the actin filament. And as the myosin component changes its conformation, pulls the actin filament along. It also then detaches from the actin, recovers to its original position before binding again, exerting the force. And we can now have a good idea of exactly what's happening here. This whole process depends critically on ATP, which finally shows its effect in the power stroke at the end. But ATP is binds into a binding site on the and the myosin. in ways that Robin characterized yesterday in terms of forming a host of different bonds that help to structure the ATP such that the gamma phosphate is removed. And in doing so, exerts a force that changes the physical conformation of this whole molecule, particularly altering the conformation of the site at which actin is bound so that it will either release the actin or bind to the actin. But it also exerts a force that is exerted all the way out to the lever arm, which is this component here, controlling which of these positions it's in. So and crucial to this is tracking the free energy that's released in the hydrolysis of ATP and how that leads to physical movements of parts of the molecule so as to engage in the various activities of moving along pulling the actin filament along. There are multiple motors of this kind. Alvaro had some examples up. The F0, F1 ATPase, which Alvaro discussed, crucial for constituting ATP in the first case from ADP plus inorganic phosphate due to the movements of the rotor that position the phosphates with respect to the ADP, such as to allow for the creation of the bond. It's all driven here by proton moving through the membrane, but this is actually the motor running in reverse. In the way it's normally thought of, the forward motion of the motor would take ATP down to ADP and drive protons through the membrane. Now, in this case, what one has is an activity in the end, the synthesis of ATP, due to the constituent components of this motor. Same story can be told about my two favorite molecular motors, two favorite molecules, dynin and kinesin, which are actually transport cargo. Cargo is shown as much larger than them, and that's in fact the case. It's often things as large as mitochondria along microtubules. In this case, by very much the same kind of story as I told with myosin, ATP hydrolysis in one location in the molecule alters the way the binding to the microtubule occurs, and also how the cargo is leveraged with respect to that. So one has a reductionist story into the constituents, but also a emergence in the fairly weak sense of emergence, that a new phenomenon, the transport of materials along axons of neurons is carried out. Now, I'm going to characterize these mechanisms as a special kind of mechanism. They are the ones that I think have been the primary focus of at least my work, and I think a fair number of the new mechanists. They're mechanisms that I'm now characterizing as using free energy to synthesize the organism, repair, which I'll make more of in a moment, or enabling it to reproduce, allowing the organism to maintain itself or its descendants as a far from equilibrium system. And I'm going to characterize these as production mechanisms. Their work goes into the tasks that the organism needs to maintain itself, procuring energy, transforming that energy into its own constitution, moving itself about in its environment, those kinds of activities. But now I'm going to shift the focus to how these production mechanisms themselves depend upon organisms. I'm going to argue that production mechanisms don't equal organisms, and it's very important to distinguish the characteristics of organisms from those of production mechanisms. In particular, putting together production mechanisms does not suffice to do the work needed by the organism. It will not, they can do all the individual pieces of the work, but they won't together result in an organism. You won't, such a collective, just a collection of mechanisms won't build itself, replicate itself, repair itself. In order for the organism to maintain itself as a system far from equilibrium, it must coordinate these activities performed by the different production mechanisms so that they're performed when and in the manner needed for the organism to maintain itself. So the idea here is mechanisms do not individually repair, construct, repair, or maintain themselves. They are constructed, maintained by other mechanisms, those already constituting the organism. Maturana and Varela gave voice to this characteristic of organisms as responsible for their own constituents in the notion of autopoiesis, organisms construct, and they give rise to descendants by the same process of construction. Rosen, who has been noted several times, has strongly emphasized the importance of repair as far from equilibrium systems. These systems are constantly subject to disintegration and need to be repaired, or components have to be replaced. Alvaro has emphasized the importance of the energetic demands and has offered a very useful way of characterizing organisms as autonomous, as systems that do all of this work by constraining flows of free energy and directing it into the work of construction, repair, and maintenance. Now, a skeptical new mechanist might say, come on, you don't really need to get all wrapped up about organisms, because it's the mechanisms, after all, that do the work of building, repairing, maintaining the organism. So isn't it sufficient that we just focus on the mechanisms that do the work and treat the organism as just a collection of it? Well, I'm arguing no, organisms are not just collections. The mechanisms can do work, but they must do the work in an orchestrated way if the organism is to maintain itself, and if then so that even the mechanisms themselves will exist and be maintained. So mechanisms must be controlled so that they perform their work under conditions that are suitable for that work, when that work is required by the organism itself, and in particular in the manner needed to perform the work that is required at a given time. Now, this is not novel thinking in any way. Lots of theorists have advanced important components of this picture, especially Claude Bernard, who is perhaps one of the most insightful mechanists in all of physiology. He developed the idea that organisms create their own internal environment, and the activities of the various components of the mechanism depend upon the conditions in the internal environment, but also serve to maintain that internal environment. He talked about it as the constancy of the internal environment. We can fuss about that, I will in just a second, but the important idea here is that it's the organisms that's maintaining the environment. Walter Cannon picked up that notion and developed it into the notion of homeostasis, and talked about various feedback mechanisms through which organisms are regulated so as to maintain themselves in a stable condition. Now, Cannon himself recognized that organisms do not always return to the same state. They need to adjust their internal state to conditions in the environment, and some other theorists have today pushed much more that we should maybe drop the notion of homeostasis and notion of other notions like allostasis. I think that's an important discussion, but it goes beyond what I can discuss here. But I want to just emphasize, mechanisms do not just perform their activities whenever their start or setup conditions are satisfied. They do not always perform the same activity. What they do depends upon not just their constitution and the available materials and energy, but how these activities are controlled. So now I need to turn to what is it to exercise control, how to understand the relation between organisms and their mechanisms. So the extra sauce that I'm arguing for are what I call control mechanisms. Control mechanisms, like other mechanisms, perform work. But what they do is they act on the production mechanisms so as to alter what work those mechanisms do. And they do so in response to measurements they make of conditions either inside the organism or outside. Now, what this involves is that control takes advantage of the fact that not all constraints are fixed. Many are flexible. And in the production mechanism, it's the flexible constraints that allow for the mechanism to do different work on different occasions as those constraints are changed, and they can be changed by the work of other mechanisms, namely the control mechanisms. Control mechanisms do what they do because of constraints and controlling and constraining flows of free energy, but in this case, they enable the control mechanism to register conditions in the organism or its environment. This is what I call measurement, following Howard Petit, and then to tailor their action in response to the measurements. It's that as the flexible constraints in the control mechanism are altered by external conditions, either in the organism or in the environment, that the control mechanism carries out different work on the production mechanism, leading it to function differently. Now, I've emphasized both production and control mechanisms perform work as a result of constraining flows of free energy, but the work performed by a control mechanism doesn't directly contribute to the activities organisms need to perform to maintain themselves. Rather, it is performed on the components of other mechanisms, altering their activity, so that the flexible constraints in control mechanisms, and the further point is the flexible constraints in the control mechanism respond to other conditions and allow for the control mechanism to impose changes on the production mechanism in light of those conditions. Now, this is brought out in a particularly dramatic way in the fact that the mechanisms I talked about earlier, the biological motors, actually have a characteristic that on their own, when there are not the relevant control mechanisms, auto-inhibit. So dynein will form this what's called the 5 configuration when there's nothing to regulate it, and it's in that position totally incapable of walking along a microtubule. But when there's components that regulate it, it opens up and becomes an active motor. Kinesin, the same story. If there's nothing acting on it to make it move, it auto-inhibits. And then when control processes operate on it, allows it to open up so that the two heads can independently move along the microtubule, carrying the cargo with it. So the work of the protein depends, emphasize here the importance of conformation that was brought up in the previous discussions. Proteins change their conformation depending upon a variety of factors, in particular on the role of the control mechanisms, and what they do depends upon this conformation. Coming back to the example of the actin-myosin mechanism, it too exhibits autoinhibition. On its own, tropomyosin, an additional molecule, actually binds on top of the sites at which calcium can bind to the actin and inhibits, at the sites at which myosin can bind to the actin, and thereby inhibits the ability of actin to participate in the binding to myosin and be pulled along. But when calcium is available, it binds to a different component, troponin, which actually then pulls the tropomyosin off of these binding sites, making them available for muscle movement. So the actin mechanism autoinhibits and has to be acted on to release it. So control mechanisms are needed then to release these activities of autoinhibition by changing the conformation so that free energy can then be constrained into the performance of work. Now, part of what's going on here is that calcium is serving as a regulator. It's available to serve as a regulator, interestingly, because on its own, it's poisonous to cells, and hence cells have developed an elaborate set of systems, including the sarcoplasmic reticulum shown here, to sequester calcium. So normally calcium is kept isolated in particular components of the cell. It can be let out briefly to trigger activities, and fortunately for the cell, it already has the mechanisms to resequester it, so it can give a brief signal that can activate the mechanism here of actin-myosin. Already gotten to that point. So what production mechanisms are doing is linking production mechanisms to the organisms so that they perform the work that the organism needs to maintain itself in the conditions in which the work is required. And hence I'm arguing organisms are not just collectives of production mechanisms. They require, in addition, control mechanisms to set the constraints in production mechanisms in appropriate ways at appropriate times, and enable the organisms to deploy their production mechanisms in the important work that they need to do for the organism. So organisms are emergent from the integration of production mechanisms and control mechanisms. Now, early in research programs, Mechanist research programs in biology, the role of control is often hidden from view because what researchers are trying to do is set up conditions in which they can study the production mechanisms themselves. And so they set up environments in which mechanisms are fully operational. They're kept in an on condition, and hence one can then use various investigative techniques to manipulate the mechanism to understand its operation. But often after researchers begin to figure out the mechanism and then go about a process like reconstituting it, trying to put the mechanism back together or put it back into the organism, they start to realize that the mechanism doesn't always do what they thought it was doing, and it actually sometimes does different things under different conditions, and that's when they start to turn their attention to control mechanisms. And once they do, this often becomes a totally consuming activity because it turns out that there are often a great number of control mechanisms operating on even single production mechanisms, many more control mechanisms than production mechanisms. And often this shows itself in diseases. Many diseases don't result from the breakdown of production mechanisms, but from disruption of their control. And hence, disease is often a way of getting insight into the control of production mechanisms. Okay, so let me turn to the last theme. How are these control mechanisms organized? Well, there's a notion of level that can be applied to production mechanisms, at least at first. You can characterize a production mechanism. I'm using Machmer-Darden-Craver terminology, operating from start to termination conditions under the control of a control mechanism that makes a measurement, produces an effect on it, but that very mechanism can itself be controlled by another mechanism. So we can start to think, well, here's two levels of control operating on the same mechanism. Maybe we can extend that neatly. And often when we think about control in outside of of science, social context, and so on, we tend to think control is normally needs to be organized hierarchically. As I've tried to sketch here, in an organization, you might have individual workers passing information up to a controller that acts on them, but that can be then passed up to a chief executive, a president that acts upon that. This idea has often been taken over into biology. You hear talk of how the cerebral cortex is the controller, or the prefrontal areas are the central executives. I find this actually rather problematic, as I'll say in just a moment, partly because there are, as I noted a moment ago, often many control mechanisms operating on the same production mechanisms. And sometimes they are not under the control of some common entity. They simply register conditions appropriate for the regulation of the mechanism and carry out their effect. And even when they are under the control of something else, this control pattern can be very, very complex. As I tried to sketch here, you start to see something that doesn't look like anymore like a hierarchy, maybe more like a network. We can get this a further complication into the story, that the processes of control that I've characterized are one of making measurements and carrying out operations on production mechanisms, but those don't need to be done at the same place. One can have a mechanism making a measurement in one place, giving rise to a signal that's passed to another mechanism that actually carries out the effect. And once you open up that space of signaling, there's lots of possibilities. You can have multiple signals feeding into one measurement device. You can have the same signal being passed to different downstream control components. And this whole system can get, there's basically no rules on how to add components into this system, except that it better work in the end. And so you, once again, start to move to something far more like a network structure of control components. Coming back to the example of muscle, muscle, the sarcomere, is where the control has to occur. Control is exercised by calcium being released by the sarcoplasmic reticulum. But then when you look at what's controlling that, it actually turns out that there are multiple components of control. I'm going to be sketchy here, but just note that it ultimately depends on activities at the cell membrane, where there are different kinds of receptors to different neurotransmitters. These, in this case, both are G-coupled protein receptors that release the G protein, which affects things like second messengers within the cell, regulating how the muscle will behave. So we've now taken the control out to at least two neurotransmitters that are exercising control over the cell. Well, that takes us to the domain of neurons. What neurons do is allow for long distance processing between a measurement component and a effector component. The neurotransmitter being released is an effector. The receptors are detection or measuring components. But these are, at this point, the action is now not electrical, but chemical at the receptors. And one can start to ask, well, what are these neurons doing? Well, Fred Kaiser has pushed a thesis that I find very attractive, that perhaps the original purpose of neurons was not to pass information from the outside to the inside of the organism, but rather was to coordinate muscles. He calls this the skin-brain hypothesis, coordination of muscles. And if you look into organisms like, well, both invertebrates and vertebrates, often the first level of control is exercised by what are called central pattern generators, which are local controllers that can both respond to the muscle itself and create rhythmic patterns. I would argue that we've had lots of local controllers, and brains are really chimera of independent control mechanisms. They constitute a heterarchy, not a hierarchy of control mechanisms, without there being any central controller. Last time to visit muscle, when you look out from the muscle to the central pattern generator and then to the rest of the brain, what's quite distinctive is that there are a host of different control processes in this system: reticular formation control, brainstem nuclei control, the hypothalamus, thalamus, basal ganglia. Yeah, up here is the neocortex. But actually, quite remarkably, work done in the later part of the 20th century in Russia showed that cats without a neocortex can behave like cats, fend for themselves, eat, groom themselves, carry out most of the activities using these structures down here. And there's no neat hierarchy between these structures. In fact, there's a fair number of interactive capacities between them. So where is unity? Well, I don't think there's any center in the living organism that controls everything else. The organism consists of multiple heterarchically organized control mechanisms. Unity is at the level of the organism as a whole. 4 billion years ago or so, an organism arose that could maintain itself far from equilibrium. The basic production mechanisms all evolved very early and have stayed in place, including those that do things like keep calcium under control. And what's happened since then is that, to use a term from computer science, in a kludge-like manner, new control components have appeared and become established in lineages when that organization was successful enough in keeping the production mechanisms working so as to maintain the organism so that it could reproduce. If we want to understand how control is organized, we're going to have it will help to look at phylogeny and at simpler control mechanisms, and then think much like in the way contemporary large-scale programs are often the product not of a systematic design, but of multiple kludges to the original program that end up creating a more complex but viable system. So let me conclude. I've emphasized that production mechanisms draw upon sources of free energy to perform the work that organisms require to maintain themselves. The key to the work is performed, that the work that is performed is in the way the components are constrain flows of free energy. Production mechanisms are themselves constructed and maintained by other production mechanisms constituting the organism. These need to be regulated to perform the work of the organism when and in the manner needed. Regulation results from control mechanisms that work on production mechanisms on the basis of measurements of conditions and that determine the work that is performed. And I've tried to indicate that control mechanisms in biology are organized heterarchically, not hierarchically, resulting in a network of control mechanisms that jointly suffice to maintain the organism, keeping the lineage alive. That I like to note that my thinking on a lot of these topics has been influenced by a host of people. Person whose picture isn't up here because he gave the previous talk is Alvaro, who has influenced both Leo Bitch, who I'll mention in a moment, and my former graduate student, Jason Winning. It was in working on Alvaro's magnificent book, Biological Autonomy, that Jason and I got interested in a lot of these issues, including Jason taking me on a forced march through Paty and Rosen and other forgotten figures in contemporary philosophy of biology. Andrew Bohagen has come since Jason and has worked with me a lot on trying to understand molecular motors and drawing upon the framework of mechanisms that Jason and I developed. COVID has a number of interesting consequences. Leonardo Bitch was supposed to be visiting me for 2 months at the beginning of COVID. Had to go home early. So instead of getting work done in 2 months, we've extended it out over a year and a half of weekly Zoom conversations, and have worked together on trying to understand, well, how to build bridges between the kind of theoretical tradition Alvaro comes from of the autonomy tradition and the new mechanists, but in particular focusing on notions of control. And it was partly due to my then postdoc, Lina Swan, who introduced me to the basal ganglia and to thinking about how subcortical mechanisms play a critical role and form a multitude of hierarchical control systems that I got pushed further and further down the road of heterarchy. So with that, I'll stop, and if I can stop sharing my slides.